Hello, my name's Cliff Holloway, the CEO of Scancell. It's my pleasure to welcome you to Scancell's AGM Presentation 2020. First of all, I'd like to draw your attention to our disclaimer notice. So the arrangements for this year's AGM are somewhat different to previous years due to the ongoing COVID-19 pandemic and government restrictions. Unfortunately, shareholders will be unable to attend the meeting in person. So to overcome that restriction, we are making this presentation available from the company's website as of Monday, 9th of November. And we're also inviting shareholders to submit questions to the board by 10 a.m. on Friday, the 13th of November. And you can utilize the email address as noted on this slide. And we'll endeavor to answer as many of those questions as possible at the meeting, which will be held at 2 p.m. on Tuesday, the 17th of November. And shareholders will be able to phone into that meeting using the instructions that will be available prior to the meeting on the morning. And they'll be able to hear the outcome of the proxy voting in relation to each of the resolutions. So we encourage all shareholders to submit their proxies in accordance with the instructions in the notice of AGM dated 21st of October 2020. And you'll also be able to hear the Q&A session. So in this presentation today, I'm going to give a quick overview, particularly in relation to the recent financing of the company. And then Dr. Sally Adams will give a presentation in relation to the development activities, in particular in relation to SCIB1, MODY1, and also the COVID-19 vaccine development. And then Professor Lindy Durrant will give an overview of the company's recent research activities. But before we do that, I'd first like to thank our shareholders for all the support over the last year, in particular in relation to the approvals of the two financing transactions in recent months, and also participation in the two open offers, both of which were significantly oversubscribed. So to put all that in context, just an overview of our development pipeline. And the first thing you'll notice on this slide, that there's a lot going on in the company for 23 people. We have SCIB1 entering phase 2 clinical development. We have COVID-19 entering the clinic, vaccine entering the clinic in 2021 for initial safety assessment in healthy volunteers. We have our MODY1 program entering the clinic in 4 separate cancer indications. So that's like 4 clinical trials in one. Again, that entering the clinic in 2021. And then we have MODY2 in preclinical development, and then a panel of potentially valuable antibody assets also in preclinical development. So collectively, all these assets could add significant value to the company. However, to realize that value, there are 2 things that we require. We need resources and we need capital. And this is particularly apparent when programs reach the clinical phase of development. Simply, clinical studies are really expensive and they require significant resources. It's vitally important that we design the most robust clinical study and have the resources to support them to get the correct result. They are, after all, the most important test of our technology. So in the middle of this year, the company embarked on a capital raise, primarily to raise capital to take forward our lead programs, SCIB1 and MODY1, through the planned clinical studies. And we completed a capital raise of £15 million in August. And that comprised of a £5 million subscription to Redmile Group, a £5 million convertible loan to Redmile, and then a £1 million convertible loan to Vulpes Life Science Fund, and then a placement of £2 million, which was significantly oversubscribed. And that included 1 million from Vulpes Life Science Fund. And then we had an open offer to qualifying shareholders of £2 million, which again was significantly oversubscribed. So we were delighted to the outcome of this particular fundraise, particularly by the participation of Redmile Group. Redmile is a large US-based specialty healthcare fund. And we were also particularly pleased with the continued support of Vulpes Life Sciences Fund. So shortly after that fundraising, Redmile approached the company as they recognized there was significant added value that could be realized given the broad utility of the company's platform technologies. So they proposed investing a further £30 million in the company to put that capital to work and to realize that value. And that capital raise comprised of a 12.1 subscription to Redmile and also a 17.9 convertible loan. And then we also incorporated an open offer of up to £3 million, which again was significantly oversubscribed. So in August, we issued the subscription placing in the open offer at 5.5p a share, and then the convertible loan in that time was 6.2p a share. And then in October, November, both the subscription and the convertible loan notes and the open offer were priced at 13p per share. So obviously this is transformational, with the gross capital of £48 million really allowed the company to now build out the R&D team and the pipeline to match the potential value that we could achieve. So when we look at the use of capital, primarily the utility of this capital was to broaden the utility of the company's Moditope, Immunobody, and Avidimab technologies, and to build the development resources behind that. So our first priority is to advance both the SCIB1 and Modi1 and the COVID-19 vaccines through their planned clinical studies. And Dr. Sally Adams will talk to that in her presentation. And then we need to initiate and advance the new and existing Immunobody and Moditope programs, such as Modi2. And then we looked at all the resources that we can build out in the company, in particular in clinical operations, so we can expedite these programs and run multiple clinical studies in parallel. And then we can build on our existing antibody portfolio to further advance the preclinical development of those assets, particularly looking at their utility as antibody drug conjugates. And this is an area of intense commercial activity right now within the industry. And we really believe that there is significant value add if we can take these antibodies further forward and to further explore their utility in this area, rather than actually transact those assets in the very early stage of development. The other area that we'll be looking at is to also supplement the £2 million Innovate UK funding the company received earlier this year to advance our COVID-19 vaccine. And then we also want to broaden out our intellectual property portfolio. So this capital will also enable us to ensure optimal development and commercial strategies going forward. So in summary, Scancell has a broad technology portfolio. And we have these platform technologies for the treatment and prevention of disease. And so our focus now is to realize value through the execution of our development plans and leading to transactions that can meet the search criteria for larger companies as they search for new assets. So 2020 has been a very difficult year for many companies and many people, and Scancell is now stronger than ever and remains committed in the fight against COVID-19 and cancer. So again, I thank our shareholders for all their support and our employees, and I look forward to updating you as we embark on this exciting journey ahead. And now I'd like to invite Dr. Sally Adams, who will give the update on our development activities. Thank you.
Hello to all our shareholders. I'd like to start the development update by talking about the SCIB1-002 trial in melanoma patients receiving the checkpoint inhibitor Keytruda. Many of you will recall from last year's AGM that we had secured an agreement from the NHS to reimburse the costs of Keytruda and that we had opened the Nottingham site for recruitment. We'd also just withdrawn our investigational new drug application in order to allow us to start study in the UK, whilst ICOR were continuing to deal with questions from the FDA. After the AGM, we were able to submit the SCIB1 IND in December of last year and then received approval in January of this year. In the meantime, we continue to try to find suitable patients for the SCIB1-002 study in Nottingham. And although several patients entered screening, unfortunately, they were ineligible for one reason or another. As recruitment was slow, we therefore decided to open some additional sites, and we selected Oxford, Mount Vernon in Hertfordshire, and the Velindre Hospital in Cardiff. But unfortunately, just as these sites were due to be initiated, the coronavirus pandemic arrived, which rather put paid to some of our plans. At the start of the UK lockdown in March, the UK regulatory agencies prioritised COVID-19 clinical trials, and they redeployed many NHS staff to frontline COVID wards. The NHS resource for cancer trials was therefore significantly reduced, with almost all ongoing cancer trials being paused and new ones being prevented from starting. Our trial was also impacted by the shortage of ICU beds, as any patient receiving Keytruda has to have an ICU bed available in the hospital in case of any serious side effects caused by the checkpoint inhibitor. There was no formal requirement to put the study on hold, and as soon as the lockdown started to ease slightly, we began conducting risk assessments with the 4 sites to allow us to restart. Part of this process involved changes to the protocol itself to reduce patient visits and to minimize any potential exposure to COVID-19 for either the patients or the hospital staff. These changes required a formal protocol amendment to be submitted to the regulators. This has been done, and as soon as we get approval, we will aim to kick off recruitment again. Of course, the potential impact of the second wave of coronavirus still looms, and timelines will to some extent be dependent on whatever NHS resources are available as the number of COVID hospital admissions continue to rise. But if we can start as scheduled, we anticipate early data to be available in the second half of next year. So let me turn to Modi-1. Those of you who were at last year's AGM will hopefully remember me talking about the fact that Modi-1 is actually made up of 3 drug substances: the 2 citrullinated fermentin peptide conjugates and the enolase 1. Each of these 3 drug substances has to be processed into a suitable drug product formulation that can be injected into patients. I also talked about the fact that the conjugates have somewhat challenging synthetic properties, primarily that each conjugate is very hydrophobic. And although this confers the ideal properties to induce highly potent T cells, it makes them very difficult to dissolve in commonly used aqueous buffers such as saline solution. So what progress have we made? Firstly, we have successfully completed the GMP bulk manufacture of all 3 drug substances, including the ENO-241, which was significantly more difficult than the 2 fermentin conjugates. However, we have made high purity batches of all 3 and in gram quantities, and these are now on long-term stability studies to determine their eventual shelf life. For the 2 fermentin products, we have successfully completed formulation studies and the GMP manufacture of more than 2,000 vials of each of them for use in further stability studies and for the clinical trial itself. The ENO-241 conjugate has again proved more challenging. However, we have identified a formulation strategy that we plan to take forward to GMP manufacture very shortly, and I'll give you a bit more detail about that in a minute. We have developed and validated analytical assays for each of the 3 conjugates, and although I know this sounds simple enough, it actually requires a lot of specific development work to refine the assays so that they are sufficiently sensitive and specific to be used for the routine assessment of both the content and purity for each of the 3 GMP products. We have conducted a formal regulatory compliant toxicity study, and this has been completed with no evidence of any local or systemic toxicities being reported. This is obviously a critical hurdle for us to overcome, and it not only supports the use of Modi-1 in the clinic, but also the intradermal route of administration and the starting dose for the clinical trial. You may also remember that we had a successful meeting with the German regulatory authorities in 2019 to discuss the Modi-1 vaccine program. We followed this with a meeting in February of this year with the MHRA in the UK, which was equally productive. The successful outcome of this meeting should allow us to have a very smooth progression of the clinical trial application once the manufacturing tasks are complete. So let me talk a little more about formulation of the Modi-1 peptide conjugates. One of the delights that we've all had to learn to live with during the pandemic is obviously the wearing of face masks. Now, you all know that the masks are there to protect others, and the way that they do that is to prevent very small particles, such as viruses and bacteria, passing through the pores in the face covering. And we use the same principle to make sure that the solutions we inject into people are safe. So when we make solutions for injection, we sterilize them by pushing them through a special filter with very tiny 0.2 micron pores in it. If there's anything bigger than this, then it won't get through. And clearly, although we want it to stop any infectious particles going through, it will also block any very small particles of product. When we took the 2 fermenting conjugates, we were able to develop a solution at the required concentration that was completely soluble. When this was put through the filter to sterilize it during the vial filling process, everything was fine, and the material at the end was still at the required concentration. However, despite trying many different solvent combinations, the ENO 241 conjugate was resolutely insoluble. So if you try to push this through a 0.2 micron filter, although you might get a clear solution at the end, most of the material sticks to the filter and there isn't going to be much left in the vial. However, I'm very pleased to say that we have now found a way forward. We have a solvent in which we can dissolve the ENO 241 conjugate. We can't use this solvent undiluted in the clinic, but we can use it to solubilize the ENO 241 and then filter sterilize the solution. Keeping everything in a sterile environment, we can then add sterile water to achieve the final concentration of drug product and also a suitable dilution of the solvent. And although there may be a slight haziness to the final product, it can still be filled into vials as a sterile solution without having to go through another filter. And again, although this might seem quite straightforward and not very different to what I was talking about on the previous slide, there are actually a number of development steps involved to ensure that the process is both robust and reproducible, all of which currently have to be contracted out to a third party, which slows our progress and requires a significant resource. But I'm delighted that we've now got to a point where we are ready to do the final GMP manufacture for ENO 241 so that we can get into the clinic as quickly as possible. Moving on to our COVID-19 vaccine, I just have a brief update about the manufacturing program. Lindy will take you through the science of the vaccine itself, but in terms of manufacturing, it is a standard plasmid DNA that has an almost identical backbone to our immunobody constructs. As we've already got an excellent safety study from the SCIB1 trial, we anticipate that the regulatory authorities will require minimal toxicity studies to be conducted prior to clinical use. And these discussions will be the subject of a fast-tracked scientific advice meeting to be held with the MHRA. As we announced recently, we've contracted Cobra Biologics to manufacture the selected plasmid, and we have already initiated GMP cell bank manufacture. GMP production and testing of the plasmid DNA is scheduled for the first half of next year, with the trial anticipated to start early in the second half of 2021. And finally, some clinical timelines, but again, with the caveat that certainly for the cancer trials in particular, these are subject to NHS resources being available in the ongoing pandemic. So let's start with SCIB1. As I said earlier, we've amended the protocol to minimize patient visits, and as soon as this is approved by the regulators, we will restart recruitment. With the additional sites on board, we're still aiming to complete recruitment within a 12-month timeframe. There will be ongoing assessments of safety and tumor status throughout the study, and as it isn't a blinded study, we therefore hope to have a rapid readout on efficacy. Next up, we have Modi-1. Believe me, the manufacturing challenges have been just as frustrating for us as they have been for you. But we have a way forward, and we anticipate being in the clinic in the first half of next year. As this is a first-in-human study, the key parameter is obviously safety, but we will also be monitoring early efficacy readouts too. We're aiming to have the dose escalation phase fully recruited during the second half of next year, and we'll then expand into the 4 expansion cohorts that we've spoken about before. And finally, the COVIDity programme. This is an incredibly tight schedule, but we are on track with the initial preparation stages for manufacturing. We're planning to take the programme forward for MHRA scientific advice within the next month or so in order to fast track the selected plasmid into the clinic. The trial itself is likely to be conducted at an accredited phase 1 unit so that we can ensure rapid recruitment of volunteers and an immunogenicity readout as soon as possible. So we have a busy year ahead, as you can see. And so with that, I will hand over to Lindy for the research update. Thank you very much.
Good morning, everyone. I'd like to add my thanks to Cliff and Sally for everybody who's supported Scancell over the last year, in particular the shareholders for their very generous support, and to the staff for their hard work. It's been a very interesting year for Scancell. We moved from our building in City Hospital to this brand new building at the University of Nottingham campus called the Biodiscovery Institute. It's a fantastic building with lots of great resource, but unfortunately in March we were locked out of the building due to the COVID pandemic. And when we have returned in July, we've had to work a very strict shift system, which involves some people going in very early and some people staying very late. And this is to make sure that we are all socially distant and that everybody stays safe. Despite that, we've done some exciting research, which I'd like to share with you now. So let's talk about immunobody initially. Probably the most big thing that we've done here is the new COVID vaccine. And part of that, we found out that by incorporating some of the FC modifications, which is our avidimab technology that was part of our monoclonal antibody work, into the vaccine, we can actually get very good T-cell responses, and we can now incorporate that modification into all our immunobody constructs and filing a new patent and thereby extending the patent date for immunobodies going forward. In terms of Moditope, Modi1 will be going into the clinic next year, which is extremely exciting. Modi2, which was our second modification known as homocitrinylation, hopefully we now have the resource to take that into the clinic, which also is incredibly exciting. Modi3 is more citrinylated epitopes, and potentially in combination with Modi1, we're trying to develop as a vaccine post-surgical resection of your cancer to prevent your tumour coming back. And hopefully, due to the very broad spectrum of expression of these antigens, we're going to get really nice expression. If we look at the monoclonal antibodies, FG129 is an antibody we link to a drug, so we use as an antibody drug conjugate, and I'll tell you a little bit more about that. Again, we are looking to see whether the avidimab technology will help other people's ADCs to become more effective. FG27 is a second antibody, that's a pure avidimab antibody, so it's a standalone antibody that punctures holes in cancer cells. And we're hoping to take that forward in gastric cancer. FL134 we're going to use as a CAR-T. So FL134 binds to what we call small cell lung cancer. There's two types of lung cancer. There's non-small cell and small cell. Small cell is about 20% of total number of lung cancer. It's an extremely aggressive disease with a very poor prognosis. So if it's possible to develop a CAR-T to treat that, that would be a significant step forward. We'll only be doing the CAR-T in the lab. We'll be verifying that it's a good target and that it works. Clearly, we'd need to do a collaboration or a deal with another company to take this forward into the clinic. Our fourth antibody is an antibody that doesn't actually target tumour cells, but targets T cells. And that means we can use it to either stimulate better T cell responses in cancer patients to give us better outcomes, or indeed we could use it in patients with autoimmune disease to actually kill T cells to reduce their overexpression or their overstimulation of T cells. Again, very much still in the lab, but very exciting. As I say, I've already mentioned the Avibimab technology. It's just where we basically change the Fc slightly of the antibodies, and that improves their avidity, or what we call potency. And we've been applying that to all our technologies, but it equally has the ability to potentially increase the avidity of any antibody, and therefore could be a possibility to license to other people. So we're looking at the moment to see how broad that platform can be used. The TCR technology is now fully developed and working. We can clone TCRs, we can clone them into virus, and then we can transduce T cells to express new TCRs. That all works beautifully. So far, we've screened 8 out of 24 MODY1 TCRs, and unfortunately, none of them have come up positive to this point. That's probably due to the fact that we've been trying to clone these responses from unimmunized normal healthy donors, and they're CD4 responses, which by their very nature are lower avidity than CD8s. So that makes this technology very challenging. We will continue to screen these TCRs. Hopefully we'll get lucky in some of the others, but if not, I think this technology may need to wait for the actual MODY1 clinical trial. And when we actually immunize patients here, we should get much stronger T cell responses, which we'll then be able to clone. In terms of using the antibodies to deliver potent drugs, what we have here is you normally have a drug that's very toxic, toxic. So if you give it systemically, it probably would kill people. But if you actually link them to an antibody, you can find that you can then use the antibody to deliver the drug directly to the inside of a cancer cell. And that's what we're showing you here. So this is the antibody here in green. This is just showing you the cell membrane of a tumor cell. And hopefully what you can see is the antibody is a little bit on the cell surface, but also intracellularly. And if we stain that with components called lysosomes, now lysosomes actually allow, they have enzymes which will actually cleave the drug from the antibody, and then the drug diffuses into the cell and kills it. And that's exactly what we've done here. What we've done here is link our antibody 129 to a variety of different chemicals. When they internalize into the cancer cell, they kill it. So that's what you're seeing here. These are irrelevant control antibodies that don't bind to the antibody. You can see some of these drugs, even attached to irrelevant antibodies, are still pretty potent, but some of them are not. And this particular drug here called auristatin, you can see has no toxicity with an irrelevant antibody, but very potent with our antibody. So we took that into what we call a xenograft model. So these are human tumors that grow in mice that don't have an immune response, so they're called nude mice. So they grow quite nicely. And you can see that's what's happening here, growing beautifully. If you attach the drug to an irrelevant conjugate, you see very poor inhibition of growth. But if you put it on our antibody, not only do you stop the tumor growing, but you completely clear the tumor from these animals. And this work's actually published in Cancer Therapeutics. So if you want to go away and read a little bit more about this, then you can do that. So then I wanted to just spend a little bit of time on the COVID vaccine, because I know that's very interesting to a lot of you. In essence, this is the COVID-19 virus, and you can see what it does is it binds by its spike protein here to this receptor called ACA2, and that's how the virus gets inside a cell. So what our vaccine needs to do is two things. The first thing is it needs to do is stimulate neutralizing antibodies which prevent the viral antibody. So these are the antibodies here. They bind here to the spike protein. It can no longer bind to its receptor, and it can no longer get inside a cell. And in order to stimulate these neutralizing antibodies against this spike protein, we've actually used just the receptor binding domain. So this is just the bit at the end here where the two molecules interact. And by doing that, we make very specific antibodies just to this domain, not anywhere else. Which focuses the immune response and gives you a much better vaccine. But the other thing we think a vaccine needs to do is stimulate T cells. You didn't think you could get away with a talk from me without mentioning T cells. T cells are my reason for living, and T cells are just as important in viral infections as they are in killing cancer cells. So what we also incorporated is the N protein, the nuclear capsid protein, to stimulate potent T cell responses. And the reason we use the nuclear capsid protein is because this is conserved among many different coronaviruses. So if you can get a T cell response that recognizes the N protein, it would kill not only COVID-19, but potentially other COVIDs as well. So that could have broader reactivity and give us much better protection. It is also possible to get T cell responses to the receptor binding domain, and they can also help with the vaccine. What was particularly rewarding, as I've already mentioned, is if we attach these proteins to FC and indeed modified FC or avidimab, we got the strongest, most potent T cell responses. And that's the basis of our new patent, and it's also the basis of the vaccine that we are going to develop going forward. As you already know, most of the DNA work we've done to date, we use an electroporation device in collaboration with ICOR. And that's great, and it works pretty well with the cancer cells. The trouble is trying to get lots of devices all around the world for a prophylactic vaccine is going to be quite challenging. So with the University of Nottingham, we've been trying to develop a non-toxic peptide delivery system, which will be as effective as electroporation, but will be just a simple injection. And we're making very nice progress with this going forward. So we made a whole variety of different constructs. So with the receptor binding domain, we had a monomer, just the protein itself, and we had a larger or a smaller complex for that. We took the monomer and linked it to FC, or we took the monomer and linked it to a trimer. And that's because the receptor binding or the spike protein on the surface of the coronavirus actually exists as a trimer. So we thought that might be the best way of stimulating neutralizing antibodies. We also made a simple dimer with just a cysteine bond, and we also, as I've already mentioned, made an FC with a modified FC fusion. Slightly simpler for the N protein, because for the N protein we just needed T cell responses, so we compared monomer, monomer linked to FC, and monomer linked to modified FC, and we made all sorts of combinations of these different things. Sorry. Clearly, the whole point of the modified FC is to target this high affinity receptor CD64 on activated dendritic cells, and that's been the basis of our immunobody platforms going forward. This is just showing you a little bit of the data. In essence, what we're showing you here, this is the responses to the RBD, and it's either to the T cell or neutralizing antibodies. Again, if you look at the T cell responses just to the RBD protein, you can see we get responses to the monomer and the trimer, but we get better responses if we link it to FC. And that's also true rather than just the small bit of the protein we're interested in, the whole S1 protein. Again, the RBD FC looks good. If we look at the avidity, for those of you who remember what avidity is about, it's the potency of a T cell. And what you can see here is you get a nice frequency response to the monomer, but as soon as the peptide drops down, we lose it. So in essence, basically activity is only there at very high peptide concentrations. Anything that moves to the left on these curves gives better avidity. So you can see the trimer is giving a stronger avidity response, but the FC is clearly the best high frequency, high avidity response. It was a slightly different story as ever when we went on to look at the virus neutralizing antibodies, because in essence what you can see here is the best one here is the monomer at giving neutralizing antibodies, although the trimer and the FC are also pretty good. So it's ultimately going to be a compromise between the highest avidity, most potent T cell response and the highest avidity, most potent antibody response. And at the moment, we're still trying to decide whether to use the monomer or indeed the RBD FC. We have a few experiments left that we're just completing. A little bit simpler with the N protein, as I said, when we compared here the T cell responses to just the monomer linked to FC wasn't a great deal of difference, good response to both, but clearly there was a significantly better response to the modified FC. Again, higher frequency, more potent responses. And that's whether we measured it against the N protein or indeed against specific CD8 T cell epitopes within that N protein. So definitely the vaccine will contain the modified FC. So finally, just again, very briefly to explain why we think the avidimab enhanced FC is so good. If you remember back to the good old days, the immunobody can go in by 2 different routes. The DNA can go directly into an antigen presenting cell, where the protein's made, it's degraded into small peptides. They're presented on MHC class 2 to stimulate helper T cells, or MHC class 1 to stimulate our killer or cytotoxic T cells. But our vaccine is given intramuscularly, so the vaccine can also go directly into the muscle. The muscle will produce the protein, and then the protein goes into the antigen presenting cells. The advantage of having it linked to FC is the FC can then bind to this high affinity FC receptor CD64. Again, then it gets internalized and processed similarly to the direct transfection. We previously showed that if you send the DNA both as a protein and also as a DNA, we get the best, highest potent T cell responses, and that still is true. The advantage we now have is because we've modified this FC, we get coalescence of the CD64 receptor on the cell surface, and that means they get internalized much more rapidly. So in essence, we shuttle more of the protein through into the APCs to give us better presentation and stronger T cell responses. And as I say, that's not also only the basis for the patent for COVID, but is actually new life for the whole of the immunobody platform, including SCIB1, SCIB2, and any other SCIBs that we might want to make. So we're in a really strong position now to take all these things forward into the clinic and work out what's the best thing and how we can best help cancer patients.
And finally, just a reminder of the email address for any questions. These need to be received by 10 o'clock this coming Friday, the 13th of November. The telephone number for you to dial into the AGM itself will be made available on the company's website on the morning of the 17th of November. Thank you for your attention, and we look forward to an exciting year ahead.